Hi everyone, welcome back to my single-cell scATAC-seq video tutorials. In this video tutorial, I'm going to show you how to plot genomic regions using the CoveragePlot function. Using this function, we can plot the frequency of TM5 integration across regions of the genome in different cell clusters. So again, we are going to use the PBMC dataset for demonstration. First, we need to set the default array as peaks. Then we can have a look how many peaks in the PBMC dataset. You can see we have more than 87,000 peaks in the dataset and 7,034 cells. Also, we can use G-ranges to see the peaks. For example, here we are going to see the first 10 peaks. So you can see the first 10 peaks are on chromosome 1. Here is the start and end region. Because we have so many peaks in the dataset, but we only want to plot the interesting peaks, I showed you in my video 4. We can use the find markers function to find differentially accessible peaks. So for today's demonstration, we still compare the CD4 naive T-cell clusters with CD14 monocyte clusters. To speed up the process for demonstration, I'm going to use high percentage peaks that presented in the CD4 naive cell clusters as 75%. And also, I'm going to use log FC threshold bigger than 2. So let's run the find markers function. You can see that's very quick. We only find 8 peaks that match our criteria. Let's have a look at the 8 peaks. You can see here are the peaks, p-value, log FC, percentage expression in T-cell cluster, and the percentage expression in monocyte cluster, the adjusted p-value. Now we can use the peak name to plot the genomic regions. For example, we are going to use the first peak. We just need to copy this peak and use the CoveragePlot function to plot the genomic regions. So you can see I already copied the peak name here in the CoveragePlot function. So the object will be PBMC. Here is the peak name. So before we run the CoveragePlot function, we can set the plotting order for the cell clusters. Let's run. So now we can plot the genomic regions for this peak. We can zoom in. So from the plotter, you can see the signal of TM5 integration along the chromatin accessibility tracks. On the tracks for each cell type, for example CD4 naive, CD4 memory, TNF5 signals have been averaged from all cells within a cell cluster. For example, in the CD4 naive cell cluster. And also we can see important information about the gene allocation. You can see here in this peak we have the BCL11B gene. And we can see the chromosome information. It is chromosome 14 and the start and the end region. So here we just plot the peak region. We can extend the plot region to upstream 40,000 base pair and to downstream 20,000 base pair. We can run it again. Let's zoom in. You can see now we add more regions on both sides of this peak. Because this peak is in the open chromatin region for gene BCL11B, we can also just plot the gene name instead of the peak. If we change the region as BCL11B, we can plot it again. So you can see now we have a plot region for the BCL11B gene, and we can see the TNF5 integration signals on the chromatin accessibility tracks. Because in the dataset we predicted the gene activities, so we can add the predicted expression level for the BCL11B gene in the plot. What we need to do is just put the features in the plot as BCL11B. We can run the plot function again. Now you can see on the right hand side we have the plot for the expression level of predicted BCL11B expression. You can see they have high expression in the CD4 and the NK cells, but the expression is very low in B cells, dendritic cells, and monocyte. So for the scATAC package, they also developed an interactive plot version for this plot. In this video demonstration, we can also try the CoverageBrowser function. We can use the surrogate object PBMC and plot the region for CD8A gene. So let's run. You can see we have the plot here. Let's pop it out to see it in the big windows. So you can see we have the plot for the CD8A gene. You can use the minus button to see more regions on both sides of CD8A gene, or you can use the plus button to zoom in into CD8 gene. We can also use those buttons to see upstream or downstream genes. If you consider this plot is interesting, you can save the plot. So you can see here, we don't have the expression level for the CD8A gene. We can use the parameters to add in. Here we can just add the CD8A, then we can update the plot. Now we can view the plot to see the expression level for the CD8A. So you can see now we have the expression level for CD8A on the right hand side. And also we can use these windows to change the plot for different genes. For example, we want to plot CD4. Let's change it to CD4 and update the plot. Now we can view it again. You can see it changed to the plot for CD4 gene. So now we can save the plot for the CD4 gene. And also we can add more gene expression plot on the right hand side. At the moment we only have CD4. We can just add CD8 in. So we can put a CD8A, CD8A. We can update the plot again. Now we can view. You can see we have the expression level for both CD4 and the CD8A gene. Now we can save the plot. So if you don't want to see more plots, we can just press the done. If you press the done button, it will save all your plot into the pin list when we start the CoverageBrowser function. So you can see in the pin list we have 3 plots. So let's have a look at the first one in the pin list. Let's zoom in. You can see the first one saved is for the CD8A plot. And let's have a look at the second one. Yeah, the second one we saved it for CD4. And the third one, the third one we saved for both CD4 and CD8. So that's it for today's demonstration. I hope my video tutorial can help your data analysis. If you haven't subscribed my channel, please subscribe it today. Thank you.